Hello students, welcome to Digital Learning DCE. Myself Dr. Manjula. In the current session, let us study about genetic recombination in bacteria. Bacterial cell reproduce sexually by means of genetic recombination which does not involve the fusion of gametes. Instead, here there is interchange of genetic material takes place. This genetic recombination is otherwise known as genetic reshuffling, which is accomplished by either conjugation, transformation or transduction. In conjugation, exchange of genetic material is brought about by physical contact between two bacterial cells. During conjugation, the bacterial cell which contain F plasmid is referred to as F plus cell and it is designated as the donor bacterial cell, whereas the bacterial cell which lacks the F plasmid is designated as recipient cell or F minus cell. At the time of conjugation, due to the presence of this F plus plasmid which contain fertility factor, and this factor codes for pili formation in bacteria. This plasmid is going to form the conjugation tube which is act as a bridge between the donor and recipient cell. At the time of conjugation, a nick has been take place in the F plasmid. One of the strand of this plasmid through the conjugation tube or sex pili move from the donor to the recipient, and inside the recipient the single stranded plasmid undergo replication to form double-stranded circular DNA molecule, so that the recipient cell becomes F plus cell again. So, in conjugation, the transfer of genetic material from the donor to the recipient takes place through conjugation tube. Coming to the second type of genetic recombination, that is transformation. Transformation is unusual and rare form of recombination observed in bacteria. Here, the segment of DNA that carries genetic information move from the donor to the recipient cell through the liquid medium in which they grow. The process of transformation was for the first time discovered by Frederick Griffith in 1928. In transformation, the bacterial cell which are growing inside a liquid medium, under certain environmental condition, the degradation of the DNA takes place and forms the smaller segments of DNA. This smaller segment of DNA released into the liquid medium where they grow, and this released DNA is ready to taken up by another recipient bacterial cell called competent bacterial cell. When the DNA is enter into the competent bacterial cell, it start integrated into the genetic material of that particular bacteria. During transformation, this free DNA released into the liquid medium is entered into the competent bacterial cell. Either it integrated with the bacterial DNA or it remained as such as plasmid inside the bacterial gene. Hence, recombination has been taken place in case of bacteria. Then coming to the third type of genetic recombination observed in case of bacteria is transduction. Transduction is a special method of genetic recombination where the genetic material is transferred from the donor cell to the recipient by means of a bacteriophage. The transformation was first discovered by Ginder and Lederberg in 1952. Here, the small piece of bacterial chromosome is incorporated into an attacking phase. When this attacking phase infects the new host cell, it injects the genetic material into the bacteria. So, let us study the steps involved in transduction one by one in detail. In the first step, when a bacterial phage come and attack the bacteria, it inject its genetic material into the bacterial cell, and at the time of replication, the bacterial DNA divided into fragments, and along with the bacterial DNA, this viral DNA also undergo replication, and this viral DNA uses the bacterial cell machinery to synthesize the virus particle. At the time of the synthesis of new daughter phage, a segment of bacterial DNA enter into this bacteriophage along with a bit of viral DNA. This phage is known as the temperate phage. When the viral particles are completely formed, the lysis of the cells takes place and releases the bacteriophages into the external environment. When the bacteriophage, which contain the bacterial DNA, is referred to as the temperate bacteriophage, when it is infect another bacterial cell, it injects its genetic material which contain both viral DNA as well as bacterial DNA. At the time of replication, this bacterial DNA released from this temperate bacteriophage integrate into the bacterial DNA present inside the bacterium. Hence, the recombination of the genetic material is observed in case of transduction, which is mainly takes place by means of a bacteriophage. To sum up the session, in bacteria, sexual reproduction takes place by genetic recombination, which does not involve the fusion of gametes, but this takes place only through exchange of genetic material, which is either accomplished by conjugation, transformation or transduction. The interchange of genetic material in conjugation is brought about by the physical contact between the two cells by the formation of a special tube called conjugation tube or sex pili. In the second method of genetic recombination, that is transformation, here the segment of free DNA that carries genetic information move from the donor cell to the recipient cell through a liquid medium in which they grow. In case of transduction, genetic material from the donor to the recipient is transferred by means of a special type of virus called bacteriophage. For further details about the current topic, you can refer the following references. With this, I am going to conclude this session. Thank you.